Hi everyone, my presentation is on pancreatic cancer and oral microbiome. So pancreatic cancer is a malignancy of the pancreas. It is a deadly and aggressive disease with an overall poor prognosis. The overall 5-year survival rate in the United States is 10.8%. There are two main types, the pancreatic ductal adenocarcinoma and the pancreatic neuroendocrine tumor. Researchers are yet to discover clear causal factors associated with pancreatic cancer, although smoking, alcohol, diabetes, and obesity has been identified as possible risk factors. Only 5 to 7% is thought to be related to genetic factors like mutations in the BRCA2 gene protein and other DNA damage repair proteins. Most cases are detected late due to ambiguous symptoms like back pain and loss of appetite, which are usually mistaken for other causes. There is a great lack of understanding of the underlying pathogenesis, as well as effective early detection and prevention of this disease. In 2018, an article was published in the British medical journal Gut that examined the link between human oral microbiome and pancreatic cancer development. This was a first of its kind study that hypothesized that oral bacteria influenced risk of pancreatic cancer through intermittent yet persistent migration of oral bacteria to the pancreas. Previous studies on pancreatic cancer identified that a history of periodontal disease and the presence of circulating antibodies to selected oral pathogens are associated with increased risk of pancreatic cancer. However, a direct correlation between oral microbiomes and pancreatic cancer have not been evaluated up till this study. This was a nested case-controlled study where cases and controls were obtained from two separate prospective cohort studies. The study provides supportive evidence that oral microbiota may play a role in the causation of pancreatic cancer. As you can see, this is the first page of the article. Let's take a look at the molecular methods used in this study. So oral wash samples of 361 cases of pancreatic adenocarcinoma and 371 matched controls from two prospective cohort studies were used to extract bacterial genomic DNA for DNA sequencing. From the extracted DNA, the 16S rRNA gene V3 to V4 regions were amplified and the amplicons were sequenced via a pyrosequencing system. Both groups were compared for baseline overall microbiota composition, carriage of known oral pathogens, and relative amounts of bacteria. The associations between oral microbiota and the risk of pancreatic cancer were then statistically analyzed using regression models. The results of this study identify that the carriage of oral pathogens Porphyromonas gingivalis and Aggregatibacter actinomycetum comitans were associated with higher risk of pancreatic cancer, and the phylum Fusobacteria and its genus Leptotrichia were associated with decreased pancreatic cancer risk. Risk related to these phylotypes remained after exclusion of cases that developed within 2 years of sample collection, reducing the likelihood of reverse causation in this prospective study. These are the statistical data published in the article. We can see here that the carriers of Porphyromonas gingivalis and Aggregatibacter actinomycetum comitans both has higher odds ratio of developing pancreatic cancer, whereas carriers of the phylum Fusobacteria and Leptotrichia has lower odds ratio of developing pancreatic cancer. So, in summary, this study has identified that the carriers of periodontal pathogens, Porphyromonas gingivalis and Aggregatibacter actinomycetum comitans, and carriers of low number of Fusobacteria and its genus Leptotrichia are associated with subsequent risk of developing pancreatic cancer. The associations are not likely due to smoking and other potential confounders. Also, although smoking and alcohol can alter the composition of the oral microbiota, this study did not find any significant association between them. The study provides supportive evidence that oral microbiota may very well play a role in the etiology of pancreatic cancer. The authors of this article believes that the mentioned oral bacteria are key culprits in the initiation of periodontal disease and tooth loss. Previous studies reported that a history of periodontal disease and tooth loss, basically poor dental health, are associated with increased risk of pancreatic cancer. The reports on association between blood antibodies to selected oral pathogens and increased risk of pancreatic cancer has also been published. Several other global studies, like the ones in Iran and China, have also revealed similar outcomes. However, underlying microbial pathogenesis and biological pathways to pancreatic cancer remain unanswered. The identification of specific oral bacteria related to pancreatic cancer will aid in the development of potential biomarkers in the future, identifications of high-risk patients, hopefully, and the development of targeted prophylactic therapies against causative periodontal pathogens, which overall would decrease the risk of pancreatic cancer. These are my references. Thank you for watching. Have a great day.